Get started now. Hello, everyone. I'm Patrick. I'm going to talk about two topics: the winner's curse and population stratification. I'm going to kind of quickly move through the winner's curse stuff and spend more of the time on population stratification because I think that's something that we all think about a bit more in the work we're doing. Is this not? Okay. Great. Okay, so who's ever heard of the man flu? Do you know what's the man flu?
It's how men complain about being sick.
Exactly. Okay, so I get a bad case of the man flu. I think I eat way too much junk food, and so like some mornings I wake up and I'm like, that's how I'll tell my husband like, John, I'm feeling sick. I think I have the flu, and he's like, you're fine. And I'm like, no, take my temperature. And so he'll say, okay, fine, he'll take my temperature. And some days it's like, 98 degrees, and he's like, see, you're fine. And some days it's like 102, and he's like, oh, yeah, you are sick. And so let's say that I'm being a scientist, and like, well, I'm curious, like, what's my average temperature when I have the flu? And so when my temperature is above maybe 100, and John tells me that, yeah, I actually am sick, I write that down for my temperature. And then over several months maybe, I have a handful of things. I take the average and I report that as like the average temperature of someone with the flu. What's the problem with that? What's the problem with that estimate that I'm going to get if I take the average across these numbers I've written down? Yeah.
You're basically selecting for when you think you have the flu.
Exactly. So like some days maybe my temperature is like 99 and I do have the flu. Like if they swabbed me and tested me, they'd find out that I do have the flu, but my temperature just happens to be a low draw. And so we're only recording the temperatures on the times that I happen to have a high temperature draw. And so our estimate of the average temperature of someone with the flu is going to be too large. And so that's this is a problem. We'll just leave it there. It's fine. Now, let's see if I can move it somewhere. Where if I drag it, it's going to like mute me. Oh no, look, we'll just put it over here. That's likely to be okay. Great. Okay. So this is just a general problem. The idea of the winner's curse is a general problem in research. So if you remember some of your statistical theory, when we do ordinary least squares, under fairly weak assumptions, ordinary least squares will produce unbiased estimates of the association between two variables. However, there's been this empirical thing that people have noticed that when you do a study and you look up the reported associations that are reported in the literature, and then you try to replicate them later, in general, the replicated effects often are smaller than what you've originally seen in the original studies. And so this general empirical result is called the winner's curse because if you succeeded at getting something published, but then you try to replicate it, you're cursed to get a lower estimate the second time. The basic reason, like the underlying principle of why this happens, is just an idea that you shouldn't use the same samples that to identify associations, or in GWAS it's snips, things like that, as you do to estimate effect sizes. It's a concept related to regression to the mean. We're going to talk a bit more about how this works. Because I dragged this, I need to click over here again. One point I also want to talk about here is winner's curse is relevant to all research. So we're going to focus on GWAS and genetic studies because that's what we're here to talk about. But winner's curse is a problem always. We should always be thinking about if we're looking for associations that exist in the same data that we're trying to estimate the effects, it's often going to overestimate the actual effects. So in GWAS, there's sort of two types of selection that happen, if you remember from Dan's lecture, that could lead to winner's curse problems. And I refer to them as selection by threshold and selection by rank. And so selection by threshold is based on our statistical significance threshold. When we're looking for lead SNPs in our GWAS, we say, well, we want to avoid finding false positives, and so we're only going to say a SNP is associated if it has a p-value that's less than 5 times 10 to the minus 8. And so, holding constant sample size and allele frequency, that's equivalent to saying we're only going to say a SNP is significant if its estimate is above a certain threshold. So it goes back to this flu fever problem that I was talking about before. And so this p-value threshold is great to correct for multiple testing. It's great to avoid type 1 error rate, but it will lead to biased estimates when we're looking at the actual betas from our GWAS coefficients. So here, I'm a visual person. I like thinking visually about these sorts of things. So here's a visual explanation of why this is. So imagine that there's a SNP where the association between the SNP and the outcome, the true estimate, if we have an infinite sample size, it has an effect of 0.1. Well, OLS is unbiased, and so this gray PDF that I've had here is the distribution of the effect sizes if we were trying to estimate an association of this size in a sample of about 100 people. So you can see it's centered at the truth, but sometimes we estimate a low, sometimes we estimate a little high. But then if we condition on being significant at 0.5 in this case, I think 0.5 probably. Yeah, 0.5. And say, what's the distribution of our effect sizes conditional on finding a significant effect? The distribution is this red thing here. So you see we've hollowed out this middle section because if it's in this, then it won't be significant. But otherwise, there's a much higher effect on the left and right. So if this is the PDF, we see a ton of weight here on the left side. And so what's going to happen to the expected estimated effect conditional on being significant in this case? Who thinks it's going to go that way? Who thinks it's going to go that way? Who's non-committal? All of you. OK, this way or this way? Hands up. I see like 3 fingers up. OK, good. Over here, right? Look at how much mass is over here to the left. And so if we were to think like, what's the expected value of a variable with this distribution, it's going to be centered around where this mass is. And so we're going to get an expected estimate of something over here relative to the truth, which is 0.01. And so this is just the inflation due to Winner's curse. This is also related to, Dan was talking in one of his earlier lectures about type M errors, magnitude errors. And so you can see that in this case, we're not very well powered, right? In fact, there's even if we estimated, if our beta estimate were equal to the truth, like we're not going to believe it because our critical values defining significance are outside of where the truth is. So this is a really underpowered study. Because we're very, very low powered, there's going to be a lot of inflation. So that's basically how Winner's curse operates through this selection procedure that we do in GWAS. Great. The next type of selection is we select by rank. So because of LD, we don't want to say that every genome-wide significant. We don't want to look at all of them because if there's a bunch of SNPs in one region, they may all be genome-wide significant even if there's only one causal SNP in the region, because any SNP that's in LD with the causal SNP, could possibly be genome-wide significant. And so we say, well, the solution there is let's just pick the SNP with the lowest p-value in the region, and we'll say that's the lead SNP. But now we're doing another selection, right? And we're selecting based on the thing that's the most significant. It's going to, the one with the highest beta. And so, so that's going to further inflate our estimates due to Winner's curse potentially. In general, I think people think a lot about this sort of inflation, selection by threshold. I don't think people talk much about the selection by rank. I don't know how, in fact, so few people have talked about it. I actually don't know how big, the bigger problem these two relative things are, but they both do exist. So what do we do about this? So I've just told you it's like, oh, well, we do these GWASs. We have betas which we use and we interpret in a lot of the stuff that we do. And I've just told you that they're all garbage, right? Or not totally garbage, but at least they're biased and inflated. And so how can we fix it? Well, one thing that we could do is just do a replication study, right? So I've told you that the source of the Winner's curse is that we're using the same data to identify SNPs as we are to estimate effect sizes. So if we had two samples, we could use maybe one set of data to identify SNPs and another data to estimate the effect sizes. And because OLS is unbiased, these estimates in the replication sample would be unbiased. So that's one thing that we could do. It's perhaps a little wasteful, though, right? Because if we want a lot of power to do our GWASs, then perhaps wasting some of our data so that we can get unbiased effect sizes, like perhaps it's more important to us to find the SNPs than it is to have unbiased estimates. So maybe this is not what we'd want to do. Another thing that we could do is maybe just correct. Like I said, the winner's curse has a predictable, it's predictable in the direction of the bias. It's always going to be biased away from 0. And so maybe there's a way for us to like deflate them back towards 0 and end up with something that's unbiased. And so there's a handful of methods that have been developed to try to do this. I'm not going to go into detail about all of them. I think that they're on the reading list if you're like really excited about this. But I am going to talk a little bit more about one of them, just using a standard Bayesian correction. And so for our Bayesian correction, we're going to use Bayes' rule. And so what we're interested in is, we have the distribution of the true effect sizes. So B is like the truth that we'd like to know, conditional on the estimated effect size and conditional on that being significant. Well, if we know the estimated effect size and it's significant, then once we know beta hat, the significance of beta hat's redundant. So we can just go to this. And then we use Bayes' rule to get to here. And so now this is we have a bunch of terms here that we know a little bit about. So this first term, f of beta hat given beta, that's the likelihood function. And it's just related to the amount of error that exists. If we know the true beta, then beta hat's going to be distributed normally around the truth. So this is something we know a lot about. And f of just beta by itself, that's the prior. And so we have to make some assumption about where these effect sizes are being drawn from. And we can do that empirically. Ultimately, we just have to make some sort of decision. You'll often see people using Gaussian or normal priors. They're often really simple and tractable, easy to work with. Not necessarily very realistic, because if we're assuming that the betas are drawn from a normal distribution, that implicitly is assuming that every single SNP in the genome is associated with the outcome. And while complex traits are polygenic, I don't know that they're that polygenic. There's potentially a lot of SNPs that just aren't associated at all with the outcome that we're interested in. And so then, some people will use things like a spike and slab prior, where you assume there's some fraction of the genome is not associated at all, and then the rest of the genome maybe follows a normal distribution. So maybe 10% of SNPs are drawn from a normal distribution for their effect sizes, but otherwise they have 0 effect exactly. And there's other kind of more complicated priors that we could use as well, but these are kind of two simple, very common examples that people use. Just a quick technical side note about the limitations of these corrections. There's this paper from 2011 where they go through and they show that no correction is perfect, that there's no correction that you could ever come up with that for all beta will give you an unbiased estimate. And so this is just to highlight that when you look at corrected estimates, if someone says, oh, I've done a winner's curse correction and the actual effect is this, like, it's not necessarily idea. It might be a little biased upward. It might be a little biased downward. The hope is that, if we look at a lot of SNPs like this, that, it's better than it was when we were had winner's curse rampant in the data that we were using. And hopefully maybe in aggregate, these biases will cancel out due to the correction. So the point of these corrections is just to get something that's a little better. Just want to highlight that turns out that nothing is perfect. It's impossible to have a perfect winner's curse correction. Yeah?
Sure. Could you go back to the last slide?
Yes.
I don't really get in practice, how do you decide on f of beta?
This one?
Yeah.
So if you don't have any data at all, then you just have to, it's a prior, right? So you can just pick it. I think that what you'll often do is use the data that you have to get the best guess possible. So I might say, let's assume that the prior is from the family of Gaussian, of normal distributions. And then we look at the distribution of the beta hats to infer what the variance of that normal distribution is. So, we can use the data to try to at least get us in the right direction to get a sort of empirical prior. But in terms of choosing between a Gaussian and a spike and slab, you just kind of have to make a choice.
It just seems a bit circular if you're using beta hat to make inference about the distribution of beta to solve an issue that comes from your beta hat in the first place.
Yeah, that's kind of the nature of all empirical Bayesian methods, right? You say, yeah, you say, well, my data kind of directs me to think about what the prior might be. The reason it maybe works a little bit better here is because we actually have a lot of betas, right? So if we just had one beta and we had to put a prior on that and we didn't really have other sources of information, then it would feel very, very, very circular. But if we're looking at a million betas, and then we're saying let's pretend that all of these are kind of related, they're maybe drawn from the same distribution, then it maybe feels a little bit less crazy to do it. But yeah, I agree. All empirical Bayesian methods feel that way.
Can you explain a bit what is inside the integral? What, in particular, what is B?
Down here?
Yeah.
Oh, so I mean, this is mostly just you can think of it as a normalization factor. I mean, like with Bayes' rule, what you have in the denominator is this should just be like, if we evaluated this integral, what it would spit out is just what is the distribution of the beta hats. But ultimately, this thing in the denominator is meant so that this whole thing is a distribution. It needs to integrate to 1. And having this thing in the denominator ensures that this thing integrates to 1. So there is an interpretation, but I think it's probably most useful to think of it as a normalization factor. Dan?
You could write it as f of beta hat. I mean, that's what you're going to get when you multiply f of beta hat given beta times f of, given b times f of b is f of beta hat comma b. And then when you integrate out with respect to b, you just get f of beta hat.
Yeah.
So that's the denominator, and that's exactly Bayes' rule. That's exactly the formula that on my slide from a couple days ago about switching the order of conditioning.
Mm-hmm. Yeah, so it is f of beta hat, but in terms of interpreting, like why is f of beta hat in the denominator? I mean, it's there because you need this to integrate to 1. Yeah.
Can I just ask an extra question? It's about selecting the snip with the smallest p-value.
Yes.
It doesn't necessarily mean it has beta, right? You just have like a small.
Yeah. No, that's definitely true. And so I've been a bit sloppy just to say like let's hold the allele frequencies and the sample sizes constant, just to make it easier to kind of think through. But it is true. If you have a SNP that has an allele frequency closer to 0, that SNP is going to have more variation, and therefore, even if it has a smaller true effect size, it may have greater significance. So that's true. Happy? Great. OK, so we did this. We did a winner's curse correction. These are results from EA1, so 2013. As Dan showed you, we had 3 genome-wide significant SNPs in that study. And so the estimates, the associations of these SNPs with educational attainment were all quite small, on the order of 0.1, even 0.01. And then if we did our Bayesian correction, assuming a normally distributed prior, this is what the expected effect size is, conditional on all these things that we were talking about. And so we can see there's just general, about a 40% shrinkage, I think, across the board. So it goes from 0.1 to 0.06, negative 0.014 to 0.009. And so then we can actually see how well did our correction do, because we had a replication sample. And so, in this particular case, the correction worked all right for this first SNP. We said, well, we think it's actually 0.06, and it turned out to be about 0.07. I guess if you round to 0.08. It's not clear that it did quite as well for these two, but this goes back again to this idea that said that no correction is, there's only 3 SNPs here, and no correction is uniformly optimal. And so if we did this correction on maybe larger numbers of SNPs, perhaps it would look a bit better. Yes?
If we could do the same thing on the replication estimates, like iteratively until the estimates is that possible to do that?
So you're saying correct these ones then?
Yeah, well, they're not necessarily, but we wouldn't be connecting these for selection
Because there's no selection.
There's no selection here. But I'm thinking of if we could, essentially like a machine learning approach, you could correct on multiple times. I don't know, use these estimates again on the discovery sample. I don't know, theoretically it doesn't make sense, but could we ultimately get to a state where we can really see the estimate?
So the question is if there's a way to kind of go back and forth between the discovery and the replication sample to try to converge to something that is less biased. I'm not seeing how that would work, given that these 3 SNPs were selected because of the discovery sample. And so if we ever go back into the discovery sample to try to correct these SNPs again, then there's already some winner's curse baked in by using those data. I mean, something that is also important to note is that the size of the bias from the winner's curse is going to be a function of how well powered your GWAS is. So if you're very low powered, if your true effects are very close to like the minimum effect that you could detect given your sample size, you're going to be correcting a lot. Whereas if we're getting to the point that we're getting a lot of data, and so a lot of the SNPs are quite distant from the critical value, which means that the correction is not that huge, at least for the largest SNPs in our largest GWASs. So I guess you could make some argument for maybe not doing a replication, because by combining both your discovery and replication sample, you're pushing yourself further into the significance threshold, and so the winner's curse, the bias from winner's curse is going to be decreased. But I think that's the only thing I can really think of related to something like that.
Yeah, I haven't seen anything that.
Yeah, I haven't either. Dan?
Just so people know that the numbers here are in units of years of schooling. Right?
Oh, yeah. Yeah, so one additional allele at this SNP is leads to an average increase in years of schooling of 0.1 years. Okay. Or I guess not 0.1 years, probably closer to 0.07 years, because these are biased.
So ultimately, the scores we will use for constructing the polygenic index are the estimates from the application stage?
No.
Okay.
No, yeah, in general, we'll talk all about this. Oh, honestly, the weights that we use are actually a bit more similar to this, because we often use Bayesian methods to create the weights, and these Bayesian methods kind of naturally do the shrinkage due to winner's curse automatically. But we'll talk a bit about that tomorrow, I think. But yeah, you could think of it as more closely resembling that. Cool. So why should we care about winner's curse? I guess it's good to know that the results are inflated. Well, one reason that winner's curse could become really important is doing power calculations. So Dan tells us you need to do power calculations, and we think, okay, well, to do a power calculation, we need to have some expectation of what kind of effect sizes we'd expect to see. And the problem is that so like let's say we're doing our we look at the GWAS results and we say, well, these are inflated upwards. And so if we use the GWAS effect estimates to do our power calculations, our betas are going to be too large, which means that we may be over-optimistic. We may think that the sample sizes that we need to replicate are smaller than what we actually need, because the true beta is lower than the beta that we've estimated. And so what you could do instead then is, if you use some sort of correction, like this Bayesian correction, to shrink your estimates, and then use that to do your power calculations, then you're going to get a more realistic view of how much power you actually have. In fact, what we actually do in EA2 and the subjective well-being paper we have here is a bit more complicated. We don't actually shrink. We look at the posterior distribution and draw from that. If you want to look more into exactly what we do, I encourage you to look in the supplement of that paper, or you can come and talk to me about it. But I don't think we have time to dive into it too deeply here. But the idea is that we are shrinking these estimates to account for the winner's curse. So Akbay et al., 2016, that's EA2. And as Dan told you, we found 74 lead SNPs in EA2. We did a winner's curse correction assuming Gaussian effect sizes. And based on those power calculations, we said that in the replication sample, we anticipate finding 40.3 SNPs that have a p-value of less than 0.05 in our replication sample. When we went into our replication sample, we actually had 52 out of 74. So we actually did even better than expected, given our power calculations. Akbay et al., this Akbay et al. Was a GWAS of subjective well-being, depressive symptoms, and neuroticism. And in this case, we did a spike and slab prior. So this is the one where we assume that some fraction of SNPs have no association, and then some fraction are drawn from a normal distribution. So in this, we had 19 lead SNPs for this paper. Based on our Winner's curse corrected power calculations, we estimated that 16.7 SNPs would have a p-value of less than 0.05. And in fact, we found 16. So this also, this seemed to work pretty well in this case. And so this is nice because sometimes when we do these, we're often asked, like, well, what do you expect to find in a replication study? Because we wouldn't expect for everything to replicate because these GWASs are quite low powered generally. But if what we're seeing is consistent with what we expect, then that's like nice evidence that what we're finding is real. Yep.
So for other phenotypes, does it seem like the spike and slab is a better correction than the Gaussian one?
I think spike and slab, so the question is, for other phenotypes, does it seem like the spike and slab is a better prior than the Gaussian? And yes, I think it is true that spike and slab is better than a Gaussian prior. However, there are other priors that are probably even better than this. It turns out that genetic effect sizes tend to have fat tails, meaning there's a handful of SNPs that have very large effects, and then a bunch that have kind of medium to small effects. Whereas if we assume that the slab part, or the spike part, I guess the slab part of the spike and slab is normal, then it just decays too quickly, and so it and as a result, it'll actually overcorrect in the tails. And so, but the spike and slab is easy to implement, and it seems to do fine. So that's what we've been using. But if you wanted something really realistic, you'd maybe want to do like a mixture of 3 normals or something where you have, one that has wide variance and one that has small variance, and then maybe also some that have exactly 0 effects. Those seem to do like even better than a simple spike and slab like the one we used here. Great. So to summarize, so if you're using the same sample to identify your SNPs as you do to estimate effect sizes, you're in general going to get effect estimates that are biased away from 0. There are some approaches that can correct for this bias that we should use if we're finding ourselves doing things like power calculations or replications of results. But none of these approaches are uniformly unbiased, and so we should just think of it as helping us improve some of the follow-up analyses on aggregates, big groups of SNPs. Yes.
And so the 3 SNPs that you showed us, the scores used for calculating the PGIs are then the Bayesian correction scores?
Not exactly. I'm saying they're more closely related to this than to this.
Yeah. I guess what I really want to know is that should we assume that particularly for educational attainment, is the polygenic index like more conservative? Like is it attributing less weight in the phenotype to genes than more? Should we assume that?
Yeah, so the question is if we should think of due to Winner's curse, if we should think of the PGIs that we produce and the weights that we're using to produce them as like too conservative or not. And I don't think so, because again, I mean, there's the other thing. The reason why I'm saying it's not any of these things is because most PGI software and approaches also account for linkage disequilibrium, and these estimates don't. And so there's a little bit of shrinkage due to Winner's curse related issues, but there's also a correction for LD that happens, and so that's why I'm saying it's most similar to this, because there is the shrinkage like these numbers have, but these haven't been corrected for LD as well. Yeah.
So it's a separate question, but when we talk about the SNPs replicating in replication studies, is it the exact same like lead SNP that's replicating, or is it another SNP in kind of that locus?
Yeah, so when we've done replication studies, what we do is we just look specifically at the SNPs that were identified in in the discovery sample. Yeah, I mean, I guess you could alternatively, you could say, well, maybe instead you should just do a full GWAS again and see if the same SNPs are popping. But that would be a bit inefficient, because ultimately what we just want to know about is, like, are these few SNPs that we found real? And you can get away with a smaller replication sample that way because, in this case, we only had 3 statistical tests in our replication sample. And so, instead of trying to get 5 times 10 to the minus 8, we only needed to do 0.05 divided by 3.
So for instance, in like EA1 to EA2, you found that 3 SNPs were also identified. Were they still the lead SNPs? Or
I was actually looking at the Manhattan plot. It didn't look like it. They were near the top. I mean, like maybe one of them was. I don't have the Manhattan plot that Dan was showing, but I think in one of the cases, the EA1 lead SNP looked like it was at the peak of the EA2 Manhattan plot. But the other 2 cases, it was like near the top, but it wasn't the top.
Okay.
And this is the reason why the selection by rank may be a problem, right? Because the lead SNP in one sample may not be the lead SNP in another, just due to chance. Yes?
Sorry, this is a bit detached. What do you do when you have like a larger beta or like a beta that like adds like a lot of variation, like explains a lot of like the variation, but it's like significantly than 90%.
So the question is, what do you do when you have a beta that has a large effect but is not?
But no statistical significance. Is there kind of like because here you're focusing more on like betas that are like statistically significant.
Mm-hmm. Yeah. So think back to Dan's lectures about power, right? So if you had a SNP that had a very large effect, but was not statistically significant, what that must mean is that your standard errors are very, very wide, which means that you're likely to have very low power. And so if I saw such a SNP, I would suspect that the effect size is hugely inflated. It would have an enormous type M magnitude, type M error. I mean, like ultimately, you can't learn much, right? If the standard errors are so wide, what that means is that we just don't know anything. Great. Okay, so let's talk about stratification, as this is what everyone loves so much. Okay, and we talked a bunch about this already in Dan's lecture. So some of this might be review, but review is great. So the classic example when we talk about population stratification is the chopstick gene. I'm going to talk about the Jell-O gene. So I'm from Utah. And in Utah, the official state snack is green Jell-O. And I fit right in. I love green Jell-O. And so imagine that I think the amount that I love Jell-O is so much that I think this can't be environmental. I must be hardwired to love Jell-O. And I want to find all of my biological Jell-O friends. And so I'm going to do a GWAS. And because I want to find the SNPs that are associated with how much a person likes Jell-O. Okay? So I want to do this GWAS, and so I'm going to get started. And let's say I approach you, and I want to hire you as my Jell-O GWAS consultant. And I say, OK, I want to do a GWAS. I've gathered a sample of 50 people, and I'm going to do this GWAS. Can you help me? What's the first thing you tell me?
50 people? Really?
Yes. What's the problem with 50 people? No power. Okay, so that's from Dan's lecture. So you need really big samples to do GWAS. And so I say, oh, you're right, 50 does sound small. So then I go out and I do this national search. I genotype 50 million people, and I do a GWAS on my 50 million SNPs, and I find that there's tons and tons of SNPs that have a p-value of less than 0.05, and I'm like, look, there is so many Jell-O SNPs. What do you tell me then?
The p-value is too loose.
Yeah, it's too small because of the winner, not because of winner's curse, because of multiple hypothesis testing. So I'm like, oh yeah, that's right. I was not paying attention to Dan's lecture. Let's look at this. Let's look at the SNPs that have p-values of less than 5 times 10 to the minus 8. And even then, in this huge sample from all across the US, I'm finding a bunch of SNPs that are associated with Jell-O consumption. What are you going to tell me about that? Stratification. Good, yeah, that's the lecture, right? So the problem is that maybe there are a bunch of SNPs that are just more common in Utah, where we consume a lot of Jell-O. And so if there are SNPs that are just more common in Utah, those SNPs are just predicting from being from Utah. They're not predicting how much you actually like Jell-O. So there's, in this particular case, you couldn't know whether this was like truly, a causal effect of the SNP, or if it's just, based on stratification. So that's the underlying idea of population stratification. So let's dive into some of the nitty-gritty. So here's the model that I have in mind when I'm, it's a simplified model of what I have in mind when I'm thinking about population stratification. It's a slight variation on the model that Dan showed you. So we still have some phenotype Y. We have a bunch of SNPs. So we'll have, J SNPs, where X are the genotypes of the SNPs. And here I have a mu. And this mu thing is meant to be some ancestry-related fixed effect. So if we had a population where there's some people from Utah and some people from New Jersey, mu would just be the effect of being from Utah or New Jersey, right? And so the mu here is just to capture sort of mean phenotypic differences between groups of people. And so we're just for simplicity throughout this, I'm going to assume that we've standardized Y. I guess I should have put tildes over the Y's and the X's because that's what we've done before. But I'm going to assume that Y and X are mean 0, have standard deviation 1. I'm going to assume that there's some LD. So genotypes may be correlated with each other. I'm going to also assume that the genotypes may be correlated with the group that a person's in, because just by chance, there are some SNPs that are more common in some groups. And so there's some correlation between the X's and the mu. And I'm going to assume that this error term, this residual, is uncorrelated with everything else. Okay? So there's the framework. So let's talk a little bit about why SNPs can be correlated with each other. I think there are two primary reasons. There are more than two reasons in actuality, but there's, oh, yeah. Dan?
Does this framework allow for the correlation between xj and xk to depend on mu?
Yeah, sure. I think so.
Okay, so it might be worth making explicit because that's the LD difference across.
Yeah. So the point that Dan's making is that it may be that in some groups, the correlation between pairs of SNPs is different. And so you might have, like in a European ancestry population, that the pair of SNPs are really in high LD, whereas in like Southeast Asian population, they're in very low LD. Great. So let's talk about why SNPs are correlated, because I think that's going to help us think about population stratification. Well, there's two primary ways that they might be. One is just due to linkage. So when SNPs are close to each other on a chromosome, they're more likely to be inherited together, right? Because they're on the same chromosome, and unless there's a recombination between them, they just come as a pair. And so we generally refer to that as linkage, and so that's going to lead to correlation between SNPs. But population structure also can create correlation between SNPs, kind of due to this idea that I was talking about before. Let's say that there's a SNP that's slightly more common in Utah, and another SNP over here that's also slightly more common in Utah. Well, both of them predict being from Utah, and so they're going to be correlated with each other. And so linkage only can happen on the same chromosome because it's about being inherited together because they're genetically close to each other. Whereas correlation due to population structure can happen genome-wide. As long as any two pairs of SNPs are predictive of being from some group, then they'll be correlated due to population structure. And so under some assumptions, we can think of these two sources of correlation as additive. And so the covariance between SNP-J and SNP-K is going to be equal to the covariance due to linkage plus the covariance due to population structure. So let's think about what a GWAS is. In a GWAS, it's, we're going to abstract away from controls. So in a simple regression, a GWAS coefficient is just the regression of Y on a single SNP. And so if we did OLS, then this GWAS coefficient is just going to be the ratio of the covariance between the phenotype and the one SNP we're interested in, divided by the variance of that SNP. We've assumed that we've standardized the SNP, so this denominator is 1, takes us to this covariance. And now I'm going to substitute the model for Y that I showed you a couple slides ago. And so Y is equal to this thing here. And then because covariance is bilinear, biadditive. I always forget what the term is, but it essentially means that you can just split out these covariances into the sums, right? Dan always has to remind me every 2 years. Is it bilinear?
Bilinear.
Bilinear. I'm learning slowly. So if we break this up, then now this covariance can be written in this way. And so we have the sum across all SNPs of the correlation between SNP pairs due to linkage times the causal effect of each SNP. And there's a term here, which is the covariance of each SNP due to population structure times the causal effect of each SNP. And then SNPs can also be correlated with the kind of population level phenotypic effects. All right, so these are all the pieces that are left. And when we talk about stratification bias, we're usually referring to these two terms here, right? So this term is, you can think of why this is especially bad, right? Because this is just due to the fact that these SNPs are correlated with environmental stuff. It's not like the causal effects that we're interested in. And here these are effects that are correlations is just due to population structure. Maybe we don't want that either. If you look at this thing here, this is actually a combination, though, still of the causal effects of lots of different SNPs. And we usually don't stress about that. We say that's not really a big problem. Can you think of why I would be less concerned with bias due to the fact that GWAS effects are aggregating SNPs together that are in linkage? Why do we care less about bias due to linkage versus bias due to population structure? Yeah.
I guess as long as the SNPs always come together, we don't really care which one is causing the effect if we're interested.
Yeah, that's exactly right. And so when we're doing GWAS, we're often just trying to identify regions of the genome that are associated with some outcome that we're interested in. And so the correlation that's due to linkage is very local, right? So if we find a genome-wide significant hit in a particular region, we can, it may not be the causal variant, but we can be pretty sure that it's close to the causal variant. It's in the right neighborhood. Whereas if there is bias due to population structure, that's anywhere, right? So if we find a genome-wide significant hit and we know that there's bias from population structure, like who knows where the causal SNP is then? It could be anywhere in the genome. Great. Okay, so I've talked about like where the bias comes from stratification. So now I'm going to talk about, oh, yes?
Is the other worry about the population structure is replication potentially? For instance, like if we are running it again in a different population, because it's actually because of like population stratification, it won't replicate, right?
Yeah. So the point that is Roble, right?
Yes.
Yeah. The point that Roble was making is that maybe there's also issues of replication. So if our effects are being driven by the population structure in the sample that we have, and then we do a GWAS in some other sample with different population, different ancestries, then it may be that these GWAS results aren't very representative of the things that we learn in another population. And so I mean that's assuming that these Bs are also constant across populations. So it may be that there's some heterogeneity in the true effect sizes, but I do think there's something to it. If we're under some circumstances, if we're not really worried about the bias so much, we do care about the transferability. We may be okay with a little bit of this kind of bias, but this kind of bias you might expect to be more heterogeneous than the heterogeneity in this term here. Yes.
So when you do the replicability test, that sample is like totally random because you just said that you try to replicate it in some other population altogether with different characteristics. So my question is, when we do replicability, the samples are like similar in characteristics.
Yes. So the question is about whether, when we're doing replication, is the assumption that the replication sample is drawn from a population with similar characteristics. And yes, that is what you have to assume. So if you looked at kind of if you did your GWAS in some population in, I don't know, like rural Iowa, and whoever is there, and then you tried to replicate in a sample from like inner Chicago, or I don't know, just maybe where you think there's different institutions and different, they face different things, then you would maybe think that like the replication would make slightly less sense because you have reason to think that there might be heterogeneity between the two groups. In general, as Dan mentioned before, when you look at the cross-cohort comparability for educational attainment at least, the genetic correlation between groups with European ancestries is about 0.7. So it's similar, but it's not perfect. And so that might be some reason if you don't see perfect replication, like maybe it's due to some of this heterogeneity.
Actually, the question I asked was about birth cohort, like year of birth.
Birth cohort would also, could also affect it, right? If your birth cohort means that you face different institutions, and maybe even the demographics of the population are slightly different, then similarly, you might expect some heterogeneity across birth cohorts. Great. How are we doing on time? 11:47. Great. Okay, so let's say we do our GWAS, and we controlled as best as we could. We tried to account for stratification as best we could, but we want to see how well we did. And so there are a handful of methods that allow us to look at our GWAS after we've run it to try to assess how bad stratification may be influencing our results. And so there are two sort of, I would say, classes of methods for this that I know about. One is using family-based data, and the other is using LD score regression. Raymond is going to talk to you all about LD score regression later today. And so I'm not going to talk about that here, but you guys can be really excited about it. So I'm going to talk a bit more first about family-based methods. So as we talked about in our initial lectures, if you controlled for parental genotypes, conditional on your parents' genotypes, your own genotypes are effectively randomly assigned, which means that things like population stratification or any type of gene-environment correlation is going to be wiped out in a design where you control for the parental genotypes. So what you might do is, so what this means in terms of our model is that if you've controlled parental genotypes, then the correlation between SNPs due to population structure, conditional on the parent genotypes, is going to be equal to 0, and so will the correlation between your genotypes and these sort of environmental factors. And so our GOS estimates then, if you remember, originally this was the equation, but I've told you that sigma sub pop is 0 and this covariance is 0. So in a family model, all you're left with is this thing here. You just have the effect of all of the SNPs and weighted by the correlation due to linkage. So that's nice. So we have this way of getting something that's a lot more like what we want. We said that this is the thing that's maybe interesting to us, and a family-based study like this gives it to us. That's great. The problem is that family-based samples are currently, they're still pretty small. They're getting bigger. There's a study, I think it's on bioRxiv already, by Howe et al., where they've done within family GWASs of a bunch of different phenotypes, and about a sample of about 100,000 people. Is that where they are? And so, this is becoming more and more possible, but they're still relatively small relative to like the million-person GWASs that are coming out for other phenotypes. So we can't easily do gene discovery within family, but we can use the family-based analyses to follow up on our population-level estimates. So one thing that we might do is a sign test. So imagine that someone tells us, like, I don't believe any of your results. I think everything you find in your GWAS is due to population stratification, and there's no effect at all. Well, if that's the case, then this beta.fam, the family-based GWAS results, like these beta J's are going to all be 0, which means that in expectation, our family GWAS is just going to give us 0 plus or minus some noise. So what that means is that the sign of a family-based GWAS is just going to be randomly assigned, 50-50 if it's positive or negative. On the other hand, if there are real effects, and this thing is not 0, then this thing is going to be in the direction of what the true effects are, and so will our population level effects, right? Because this thing here is going to be positive, and this thing is going to be who knows what it is, right? And so if under the null hypothesis that everything is stratification, then we would expect the signs to match in our population GWAS and our family-based GWAS 50% of the time. And so we can just test that. We can just compare the signs of our population GWAS to the signs of our family-based GWAS. And if we see that they align more than 50% of the time, then we can reject the null hypothesis that our results are driven 100% by population stratification. So that's a pretty, it's a pretty weak claim to begin, I don't, some people used to believe that it was 100% stratification, which is why we did these tests and we could strongly show, no, there is something really going on here. But in terms of how big are the biases due to stratification, that's a different question. So if you're willing to say yeah, maybe there's something going on, but you're just hugely inflated by stratification. And that's the point of these regression tests that we did and done in some of our papers. So the idea here is imagine taking our betas from our family regression and regressing them onto the betas from a population level GWAS. Well, our population level GWAS you could think of breaking it into 3 components. You have the part that's due to the true causal effects. You have one part that's due to bias, due to stratification, and you have another piece that's just due to estimation error. And so in this regression, what we'd end up with is in the numerator, we would have the variance due to true causal effects, because that's the only thing that exists in the family-based GWAS. Well, there's also error in the family-based GWAS, but that error should be uncorrelated with the error from the population GWAS if the samples are non-overlapping. Right? So in the numerator, all you have is the variance here. And the denominator, we have the variance due to these 3 components. Well, the variance due to sampling, we actually can know what that is. The variance due to sampling is represented by our standard errors. And so if we just square our standard errors, then we can subtract them out of the denominator here. And so now what we're left with, once we subtract that out of the denominator, is the variance due to true effects divided by the variance due to true effects plus the variance due to bias. And so imagine that there were no bias. That the bias due to stratification was equal to 0. Then this term is equal to 0. And so when we take this, we divide this and we get a 1. And so, so we can interpret this quantity here as it means 100% of the signal is due to true effects. On the other hand, if there was a lot of bias and bias was primarily driving our results, then this thing would be very large in the denominator, which means that this whole term would be very small. And so it means that what we can, when we do this regression test, we can interpret the results as what fraction of the signal that we're seeing in our population GWAS is due to real stuff versus stratification. So this is something that a person could do, and that we do in did we do it in EA2? I don't know if we did in EA3 or not. So ideally, what we would use is parental genotypes, right? That's I think in the applications that we've done, when we did this in EA2, we were doing sibling fixed effects. Yeah, and it's also, it has stronger assumptions. But going back to kind of what Dan was saying before, now that we have we talked about Alex's imputation method? I don't remember if we've talked about that at all yet. Oh, well, you'll learn all about this. It's great. If you have siblings, there is a way that Alex worked out that allows you to impute the parental genotypes even if you don't observe the parents. And so anytime you have sibling data, you can impute parental data and always do a study controlling for parental genotypes instead of family fixed effects. And so that's what you should all do. But that's not what we've done in the past because Alex only came around recently and developed this. So this is kind of new, cutting edge stuff. Great. OK, so this is one way that you can test to see if there is stratification in your GWAS results. But maybe we don't want to actually just see if it's there. We want to do things that correct for correct for these results. Better to solve a problem than to see if a problem is there, right? And so in terms of ways of accounting for the stratification that may exist in our results, there's sort of, again, I always break things into two types of things. So in this case, there's also two types of things, two ways to account for stratification. One are sets of approaches that adjust the standard errors in your GWAS effects. And others there are ones that actually affect the, your effect sizes that you're estimating. So let's go through each of these. So let's first talk about why we would want to inflate standard errors anyways. Why would we even think this is a good idea? And in order to do this, we need to think about what it is that a standard error is supposed to capture, right? I think that intuitively, when we think about what a standard error is supposed to be, the idea that it evokes is, well, we want it to tell us kind of how much of the signal that we see is due to things that we don't care about. And in just a standard regression, the thing that we don't care about is noise due to sampling, So it's just estimation error. And so traditionally, standard errors are just capturing how much variation there is in our estimate due to the estimation error. But you could imagine wanting your standard error to also capture how much of the variation there is in your estimates due to biases. And so, so the idea behind these inflation approaches is saying, well, how much would we have to increase the standard error so that it not only reflects how much variation there is due to sampling, but it also reflects how much variation there is due to bias. And so, then our type 1 error rate will only get a significant result if 5% of the time if what's driving the SNP is sampling and biases. So that's the idea. And the first implementation, the way this was done, was through something called genomic control. This is not something that's done anymore, but I'm going to talk about it now because it was done a lot, and so you'll probably see it in the literature as you're looking at old papers. But the idea of genomic control is let's assume that that some phenotype is not incredibly polygenic. That maybe there's only a handful of SNPs that are associated. In fact, let's assume that less than half of SNPs are actually associated with the phenotype. Well, if less than half of SNPs are associated with, truly associated, right? If less than half of SNPs are truly associated with a phenotype, then if you looked at the median SNP, the SNP with the median chi-squared statistic, then that SNP should be a null SNP, right? And so if we look at that SNP, though, and that SNP isn't null, and it has a chi-squared statistics that's greater than the median of a chi-squared distribution, then the reason it's inflated is it could only be because of population stratification, bias from population stratification. Again, this is under the assumption that less than half the SNPs are actually associated. And so in that case, what you could do is to say, well, let's deflate our chi-squared statistics, and we'll do this by inflating the standard errors, such that the median SNP has a chi-squared statistic equal to the median of a chi-squared distribution. And that way, we're saying, okay, the amount of stratification of that SNP is approximately equal to the amount of stratification in every SNP in the genome. And if we just inflate the standard errors of all the SNPs, then that should capture the variation that's due to stratification. And so people thought that was a pretty good idea. What's the problem with that idea for the types of phenotypes that we're looking at?
That they're variable incentive?
Yeah. So the key assumption that I've been talking about this whole time is we have to assume that fewer than half of SNPs are associated with the phenotype. And so if it's actually the case that SNPs are very polygenic and more than half of SNPs are associated with the phenotype, well, then we would expect to see the chi-squared statistic of the median SNP to be greater than the median of a chi-squared distribution because there's a real effect there that we're capturing. It's not just stratification. And so if we're looking at some more simple trait where there's just a handful of genes that we think are implicated, then maybe genomic control is not a bad idea. But if we're looking at something like educational attainment, where we think that a huge portion of the genome is actually associated, you may not want to do it. And so so for example, how much time do we have? We have time. So this is a QQ plot. For those of you who haven't seen QQ plots before, every dot here corresponds to a single SNP. and the y-axis is the p-value associated with that SNP. And the x-axis here is the p-value that that SNP would have in expectation if the SNP were actually null. And so if every SNP in the genome were null, then it would follow, this is a 45-degree line, just in case you can't tell. You just look at the scale of these axes. But this is the y equals x line here. And so if every SNP were actually null, all of these SNPs would fall along this line here. But the reason that people thought maybe there was a lot of stratification is because for height, for example, they saw a huge liftoff of this 45-degree line, and they're like, no way is every SNP associated with height. I think we've learned that not every SNP, but a lot of SNPs are associated with height, and so I think that they kind of misinterpreted the evidence based on our kind of preconceived notions of what genetic architecture would look like. And it turns out that for like all complex traits, these QQ plots start looking like this when you get large enough sample sizes. Oh, this was just another piece of evidence here. And so what they plotted here, this is again for height, and each dot here corresponds to a chromosome, and they estimate how much of the variance of height is explained by the SNPs on chromosome 22, and they look at how big chromosome 22 is. And so if a trait is very polygenic, then you would expect that the amount of variation explained by the SNPs on a chromosome would be roughly proportional to the number of SNPs on that chromosome. And that's just what they found here. And so kind of this linear relationship between chromosome size and variation explained by SNPs on the chromosome was strong evidence that height is actually really polygenic. And this really changed kind of the way that people thought about complex traits.
Something like height, like that is different by race. Like, would you actually want to control for population stratification? Just can you explain why, like, say Dutch people are taller because they're Dutch?
Yeah. So I mean, so even though height is quite heritable, there are still a lot of environmental things that can influence height. And so if you see large differences in height between groups of people who have very different ancestries, I think just looking at the GWAS, you can't know if that's being driven by genetic differences between groups or perhaps different environmental differences between the populations. I mean, there's a lot of research about height in particular, about how much of differences between groups of people are actually genetic or not. So I'm not an expert on that literature, but I mean, even for something like height, you still can't know how much is environmental versus genetic. And so if you're looking at population differences, then you still might make mistakes by looking at it. In fact, I mean, I'm going to talk about a paper in a little bit where they had the same thinking about like, oh well, stratification is probably small, especially once you've controlled for PCs, and they found out later that actually there still was a substantial amount of stratification in the height GWAS results. So we'll look at that in a minute. Great. So we just talked about inflating the standard errors, right? The problem with that, though, is that's good for looking at individual SNP level things, but maybe we just want to have estimates that are unbiased. Like maybe we want our betas to be right. And so let's talk about the strategies that you might do to actually have better beta estimates. And so the biggest thing that that you see a lot are sample restrictions. And so just due to data availability, a lot of the research that currently exists is in individuals with European ancestries. And so there's the sense that people who are from the European continent, who are most similar to reference samples of people drawn from the European continent, are related enough. They're homogeneous enough that we can study them together, and population stratification is going to not be that big a deal. And so that's the rationale for restricting to groups that are somewhat homogeneous. So you'll see a GWAS in European ancestry individuals, and maybe another one in East Asian ancestry, and whatever groups where there are data available. Even within these groups, though, there still is stratification. So for example, we look at European samples and we say like, oh yeah, like it's homogeneous, they're European. But even within Europe, there still is stratification. So for example, there's some north-south stratification in Europe. And the allele frequencies of people who are born in like Sweden are different than those who are born in Italy. Even within the UK, there's some north-south stratification. So we're getting to be quite homogeneous. Just people with European ancestries living in the UK, and still there's some stratification that we need to deal with. One other point that I want to make about this too is that so when we first started doing GWAS, the first SNPs that you're going to find are SNPs with the largest effect sizes in general, with the caveat that the ones that are largest effect sizes and also common SNPs, so you're well powered enough to find them, right? But we're increasing our sample sizes more and more and more and more and more, right? And so now we're getting well powered to find SNPs that have pretty small effect sizes. Under the assumption that biases due to population stratification, though, are sort of evenly spread across the genome, what this means is that the initial SNPs that we found, even if those SNPs did not have a whole lot of bias due to population stratification relative to the magnitude of the effects that we were measuring, now we're measuring effects that are very small. And so a small amount of bias for those SNPs might be large relative to the effect that we're measuring. And so, I think that initially there was a lot of talk about, oh yeah, stratification is not a huge deal. It's like it's small relative to the effects that we're seeing. And that may have been true for the first sets of effects that we're finding. But now that we're seeing really large samples and we're detecting really small effects, and we're developing methods that use, like polygenic indexes, which use SNPs from all over the genome, or there's methods for like detecting, methods for detecting things like selection in the population that use effects from across the genome. Like, it may be that the amount of stratification there, which use these SNPs that have small effects, it may be the stratification there is too big for those. And so we need to be aware that depending on the application, we may be comfortable with different levels of stratification that exist. Great. So let's talk about principal components. So this is, I think, I think the most common thing people think about when they're saying, oh, let's control for stratification is let's restrict to a sample. And the next thing people say is like, oh, let's control for PCs, because that'll solve all of our problems in life. So let's talk about why they might do that and why they might not do that. So here's our model. Remember, we have the phenotype. It's the effect of some genes. And then there's this thing that's capturing sort of ancestry-related mean differences between groups. So if we knew everyone's ancestries in our data, then we could maybe just include that as a control variable, and that would maybe account for the bias, because a person's ancestry, if we had like indicators for the groups that people are from, we could include that as an indicator variable, and that would perfectly absorb this from here, and it would remove the bias from stratification that may exist. And so that would be great. Well, if we think about what information is coded in our genes, like absent anything about phenotypes and stuff, like the primary thing that our genes tell us is where we're from, who our parents are and who our grandparents are, and like overall, what ancestries do we have? Principal components, without going super deep into what they're doing exactly, are designed to capture the major sources of variation that exist in some set of variables. And so given that we think that our ancestries are the primary thing that our genes capture, and principal components are meant to capture the thing that explains the most variation, that's sort of the intuitive rationale for why we might want to use PCs to control for ancestry. And so it's a bit hand-wavy. There are like simple population genetic models that you can use to justify it a bit more rigorously. But that's the basic idea of what PCs are trying to do when we throw them in models. Because the idea is that if we have PCs, and the PCs describe our ancestries, and ancestries are sufficient to control for this omitted mu term in our GWAS, then including the PCs are going to also predict this mu, and it can remove the biases. It's a super, super fuzzy, but hopefully getting at this idea of what the goal is. All right? Okay, and in practice, this seems to work pretty. I love this figure. Andrea had it up for a little bit, but just to dwell on it a bit more, right? If you took a bunch of data from people from across Europe, and you took the first two principal components and then plotted PC1 against PC2, like what you end up with is something that looks like a map of Europe, right? You have like Spain over here, and here's Great Britain, or is that what that is? IE? Oh no, it's Ireland. Yeah, and so the first two principal components, at least, seem pretty good at capturing the broad kind of continental distribution of individuals within Europe if you have a sample like this. That's really cool. Yeah?
Has anyone tried to do this in a non-European sample?
I presume so, and given that I've not seen the pictures in other samples, I don't know how good it looks in that case.
Got it.
I know that like in the HRS, like one of the higher level PCs, like 3 or 4 or something like that, is like Hispanic, and so it wouldn't necessarily be as quite as geographic. So, but Europe, it looks really beautiful. Yes.
I think that they have a visualization of this in that Barbara Mills and colleagues textbook that over there. It's the one with the black textbook. It was on our reading list, but anyways, I think there's the figures in there.
Okay.
In different samples.
In different samples, yeah, or
Yeah.
different ancestral groups. Yeah.
I mean, the people in this sample were also like pretty highly curated, right? Like I think they were trying to find people who, going back several generations, lived in a very particular part of Europe. Whereas, like again, if you took just like an actual sample of people, like the HRS, like those are complicated people who have complicated ancestries. Where they tried to find people with very simple ancestries to make this figure, to make it look so nice.
I've seen PCAs in a worldwide scale. Like you did with 1,000 genomes, you'll get like a worldwide.
Yeah. I mean, you even in other samples, you'll see nice clusters. It does a pretty good job of clustering out continental populations, but they don't. You couldn't like lay it on top of a map like you can in this picture.
Yeah.
Great. So let's talk about how well PCs work. Okay. And let's also talk about how well PCs work relative to just controlling for something like self-reported race. And so this is a particular SNP. A gene, where the SNP is actually RS37, that thing there, but it's on this particular gene. And we're going to look at its association with educational attainment in the health and retirement study. And so let's walk through this figure here. And so first, if we just did a regression, regressing educational attainment on this SNP, and all we controlled for were sex and age, then you get this like really big, highly significant effect. And so we're like, yeah, great. But then we think, oh, wait, population stratification. Patrick told me we need to worry about this. And we have this nice measure of race. And so let's add some race indicators into our analysis and control for race. And we see that this point here is the same regression, but controlling for self-reported racial indicators. And we see there's a pretty big drop. We're like, oh, wow, there's some stratification. However, there's still a big effect here. Like even controlling for race, we see a thing. So maybe this is real. But then you're like, oh, wait, but let's actually control for PCs. That's more state of the art. Maybe it's better at controlling for ancestry than just a self-reported race. And so all the rest of these points are adding more and more PCs. And so this first one is controlling for 1 PC, controlling for 2, 3, 4, all the way up to 20. And you can see pretty quickly for this particular SNP, like once you add PCs, you lose all of the signal. And it's suggesting that the whole effect that we were seeing over here is driven by ancestry things, and also importantly showing that just controlling for race is insufficient. Like ancestry is different than race, and the thing that is important to control for is ancestry. And so that's what these that's the first thing that we should learn. Yeah.
About the question, how many PCs are enough to add?
That's a great question. Can I come back to it in just a little bit? Okay, great. So let's say that we said, okay, I know that having a diverse sample and is maybe causing you problems. Let's just restrict to individuals who are self-reported white, because maybe it's now a homogeneous sample. Maybe that's sufficient. If you do that, and this is the estimate if you just look at people who self-report as white in the health and retirement study, and you still see a really big effect. Even controlling for 1 PC among that individual, it switches sign, but it is statistically significant. And so it's not until you start having more PCs that you start seeing that this effect is not really there in the health and retirement study. Yeah.
Yeah, so I see that you've been adding up to 20 PCs. I know this is standard in zeros now. I was wondering, regarding the discussion we had earlier, maybe this would be a problem with overfitting. I know it isn't, but I just want to know why. Like, if you keep adding PCs, maybe that would be a problem because, like, the assumption is any ancestry-related difference is due to stratification, which it probably is due to cultural stuff. But what if it's actually because of different alleles that exist in different populations?
Yeah.
Wouldn't that be a problem?
Yeah. So the question is, once we start getting up to 20 PCs, maybe there's issues with overfitting. We talked about this a little bit yesterday. So it's possible. So I'm going to say it's possible. The reason I think that it's unlikely is that these PCs. So a PC is a weighted sum of a person's genotypes, right? That's just like technically, if you were constructing a PC, the way that you would do it is you would calculate what the weights are associated with every SNP, and then so a PC you can sort of think of it as a polygenic index itself, and in general, at least for the first several PCs, the weights associated with each SNP are pretty diffuse, and so you don't have so if it were the case that like there was a lot of weight being put on the particular SNP that you were testing, then yeah, you might run into overfitting problems. And also, obviously, if you had as many PCs as you had SNPs, then that would absorb all of the genetic variation, and so there would be overfitting there as well. But given how many SNPs there are, and in general how diffuse the loadings are on each SNP for these PCs, my gut is that overfitting is not a huge problem in practice.
What if the trait was lactose tolerant?
So things that are monogenic and highly predictive. This is the point that Dan raised yesterday. So if it's something where it's a monogenic thing and highly predictive of the group that you're in, then that could cause some problems. It would more cause power problems though than bias problems, right? When we think of overfitting, I think we're worried about additional bias being introduced due to colliders and things like that. But in this case, it's a collinearity thing where the PC would be perfectly correlated with the SNP. Grant?
From Moeen, what evidence do we have that ancestry is affecting phenotype through correlation with the environment and not directly?
Not much, I don't think. I think intuitively we know that there are big environmental differences. Do you have thoughts? I think that we just don't. The big issue is that GWAS is uninformative about that question. You can't know how much is being driven by environmental differences between ancestries versus genetic differences between ancestries. I don't know. Do you have other thoughts, Dan?
No.
Dan has no other thoughts about it.
A lot of the same issues still apply, even if the source of the mean difference between the subpopulations is genetic in origin. Most SNPs are not causal, but they're differentiated between subpopulations due to genetic drift. So even if the origin is genetic for the differences between groups, you still have confounding because of that between the subpopulations.
Yeah, that actually goes back to, let's see, I have to go back quite far. Okay, so remembering this, actually no, let's go here. So this source of bias is bias due to phenotypic environmental differences, but there still is bias that may exist due to genetic differences between the groups, and that's what lives in this term here. So even if there weren't phenotypic differences, there still would be bias due to that induced by long-range LD and things like that. Yeah, Tammy?
Given that race and ancestry capture different things, is there a scenario, for example, for certain social outcomes that you would want to control for
both? I could imagine there being a scenario in which you'd maybe want to control for both race and ancestry. The reason why it's a bit tricky is because using current GWAS methods and even polygenic index methods, it's important to control, like to restrict your sample to a sample that's approximately homogeneous. And so, once you restrict to a homogeneous sample, then there's not going to be really a lot of racial variation within your sample, right? I actually don't know what the number is, but if you restricted yourself just to people who are classified as having European ancestry based on kind of these methods that we use to group people, I don't think that there would be any variation in self-reported race among that group. There might be, I guess, if you looked at a sample that was classified as having African ancestries, but yeah, I'm not sure. So the answer is like, yeah, I can imagine a design where you might want to do it, but in practice, based on current methods, due to sample restrictions, I can't think of how that would work practically. Okay, great. So race is different than ancestry, and ancestry is what's actually important in these studies. That's the takeaway here. Okay, so here's a little bit what I was talking about. I think it was based on your question, where principal components don't always they don't always work perfectly, and so there are two examples of papers where they showed that even controlling for PCs weren't sufficient to account for all of the stratification. So the first one were two papers that came out in the same issue of eLife that separately, but at the same time, they were analyzing the GWAS summary statistics from the GIANT consortium. It's a GWAS, this is a consortium that studies height and BMI. And so they were looking at the height summary statistics. And as they were searching it, they ran a bunch of tests that are meant to look at how much stratification there is. And they found that there was actually still a lot of population stratification in the GWAS summary statistics for height, despite them doing things like controlling for PCs and restricting to individuals with European ancestries and things like that. There's another paper by Abdullawi et al. Where they constructed polygenic indexes for educational attainment in the UK. And they found that individuals who are from traditional coal mining regions within the UK, on average, tended to have lower polygenic indexes even after you controlled for PCs. And so here's visually from that paper, because I think it's a bit striking. So on the left here, this is a heat map of the polygenic indexes for educational attainment by different regions within the UK. And so this is just this, the raw mean levels of the polygenic index. And you can see these areas outlined in black are areas that they've classified as being traditionally coal mining regions. And you can see within these black areas, they tend to be lighter colored, meaning that they have lower polygenic indexes on averages in that area. And so then they controlled for educational attainment with 100 principal components, and they saw the same pattern. Despite controlling for a ton of PCs, they still saw lower average polygenic scores in these regions. And so like if we group them all together, these two bars are people from non-coal mining regions, and these two bars are individuals from the coal mining regions. Just a huge difference between these two groups. And so population PCs were not sufficient to account for these regional differences within the UK.
Is this because they are moving out of those those with high polygenic scores for educational attainment are moving out of those coal mining regions?
Yeah, that's a great question. So the question is the reason for this because people who have high educational attainment polygenic indexes are moving out of the region? And yeah, maybe. I mean, I don't know that the dividing it by people who are moving away and moving towards, I think, was trying to get at that idea. But there's one question of, if a person has a high polygenic index and therefore they move out of these regions, that's actually, that's a causal effect, right? Depending on where they move. But if your parents have a high polygenic index and they move out of the region and then you're born in a different part of the UK and therefore you face a different social environment, that we'd think of that as stratification because that's not the effect of your own genes, And then if you think about maybe your grandparents did, and all of a sudden, that's it becomes a bit tricky. So we talked a little bit about this, I guess, already, but so why were PCs not successful at controlling for bias? Well, there's a bunch of reasons why you might think that. These studies, the educational attainment GWAS and the height GWAS are generally based on large numbers of studies that they then meta-analyze together. And small studies are very difficult to quality control because, it might be that your sample is too small to detect any errors that may exist. And so you have a bunch of small studies that have errors that you can't see, and then you meta-analyze them, and it compounds these errors all together. And so it may be that there is some problem with the QC pipelines in the small studies. It also could be, they did genomic control in at least Wood et al., the height GWAS, and genomic control only expands standard errors. It doesn't actually correct the betas. And so, there might be systematic errors in your in the estimates that you're using and then you're doing follow-up work on. And it may be that these systematic errors are really, in aggregate, they cause problems for the follow-up analyses that you're doing. Another issue is that PCs are really good at identifying kind of long-run continental structure, things that happened a long time ago in groups that have been separate for a very long time. But it's not quite as good at identifying kind of recent structure. And this is what I think might be happening, for example, with the movement out of these coal mining regions, right? Like if the stratification only occurred 1 or 2 generations ago, I don't know that it's been long enough for the PCs to really pick them up very well. You'd have to get quite deep in the PCs before you'd see it, I think. And then the last reason is that many of these studies were quite small. And so you construct PCs based on the data that you have. And so they constructed the PCs within their own data sets. And it turns out that if your sample is very small, then your PCs contain very little and sometimes even no information. And there's this really cute study that it's unpublished. I talk to him like every 6 months saying, when is this paper coming out? But he shared this figure with me. What this is they said, imagine you have a sample. Okay, first, imagine you had an infinite sample, and you constructed PCs in your infinite sample from a particular population, right? And so I'd say these are like the true PCs, the PCs that you get in an infinite sample. But we don't have an infinite sample, and so instead, we have to estimate our PCs in the sample that we have available. And how correlated are your empirical PCs with these like true optimal PCs that we would construct in an infinite sample? And so on the x-axis, this is the sample size of the sample you're using to construct the PCs. This is in hundreds, and so it goes from 400 to 2,000. And the y-axis is the correlation between empirical PC1 and true PC1. And so we see that PC1 is pretty good in the simulations that they've done. Like, once you have samples of maybe 1,200 people, you have a really high correlation between the true PC and the PC that you're using. But once you get to PC2, even in a sample of like 2,000 individuals, there's only a 0.3 correlation between the PC that you would like to use and the PC that you're actually using. And then once you're at PC3, it's quite small up here. And then down here where it looks like it's 0, one of the really cool results that they show in this paper is that these things that look like they're about 0 are actually exactly 0. That PCs undergo this phase transition. And that until you have a large enough sample size, higher level PCs are totally uninformative. They're just noise. And so you'll sometimes see papers where they say like, oh, we really wanted to account for population stratification, and so we controlled for 100 PCs, thinking that like, the 100th one is noisy, but like, better to include that than to include nothing. But the conclusion of this paper is that unless your sample size is very large, actually you're better off to include nothing. The PC is just noise. It's doing nothing for you at all, except potentially leading to overfitting problems, and reducing degrees of freedom. And so, in some of these initial studies where you have very small studies contributing, they're controlling for PCs, but their PCs aren't necessarily very informative about ancestry. Yeah?
Is there a reason that like the SSGAC hasn't constructed true PCs for like every person in this data combining all the samples?
So the question is, why hasn't the SSGAC created true PCs for everybody? So there are some people who what they do is they kind of do a dual approach. I don't know why the SSGAC would do it anyways, but they do a dual approach where they'll take large samples of diverse individuals, big samples, and create, make the PCs in that sample, and hopefully, so I think that's the idea of like making sure, think about what the weights are associated with each SNP to construct PCs that are very informative. And so they include those PCs. The problem with that is that if, the true PCs are population and like sample dependent, right? This population that you're drawing your sample from. And so it may be that the population that the PCs were constructed in is not very representative of the population that your data come from. And so, you have the advantage of having greater power in the loadings, but the disadvantage of maybe they're not being exactly the right PCs. But then you could also create PCs within your data. It's like maybe you should include 2 sets of PCs. I don't know of people who actually do this, but I hear people talk about things like that. Yeah.
I have a question. What variables normally go into like a sample PC?
What variables go into a sample PC?
To be able to like see like ancestry.
Yeah. So PCs are constructed just from the genotypes. So yeah, you usually don't include any other information.
Like a PC with like thousands of variables.
Yeah. So like, I mean, one could, but in practice, when people talk about using genomic PCs, they just take the PCs of the genetic data. Yeah.
So in a study that only uses PGI or PGS, and just using that maybe to see interaction effects between like an environment type or a genetic predisposition, would you say like using a PC would help you in controlling for stratification?
Yeah, so the question is, if you're doing a study of, using polygenic indexes, do you still need to use PCs to control for stratification in a study like that? Is that right? Yeah, and the answer is yes, because you still, because of the differences in allele frequencies across maybe the different populations in your sample, you may find that there are mean differences in your PGI across these different groups that are just due to chance. In the same way that you have differences in allele frequencies between SNPs that are just due to chance. And so if you don't control for PCs or stratification in some way, then if there are also differences in environment between these groups, then your PGI may be picking up just the effect of these different environments and not actually the genetic effects that we think that they should be picking up, that we want them to be picking up in most studies. I'm like way over time. Okay, I'm going to do this in 30 seconds. Okay, because another classic way to account for ancestry is through linear mixed models. And so the way I like to think about these, so one thing I just want to also highlight before is in the statistics literature and also in the econometrics literature, they use the word linear mixed models in a different way than it's used in the genetics literature. I spent a lot of time very confused about thinking that they're talking about the same thing, but they have the same name, but they're referring to different stuff. In the genetics literature, when people talk about linear mixed models, you can think in practice of it meaning, okay, let me just take an aside real quick. So if you want to think about what PCs are doing, imagine that you take your PCs and you residualize the phenotype on just the PCs, and you residualize your genotype with just your PCs, and then you regress the residuals on the residuals. So by the Frisch-Vaug theorem, that's equivalent to just including them as controls. And so if you think about, and so this residualization step you can think of as a prediction. I'm predicting a person's phenotype just using the PCs, and the idea is that residualization is absorbing any of the stuff that we don't want, anything that's like phenotypic stratification. And so, if we're residualizing using OLS, that might not be optimal. Maybe we want to use like all genotypes, and maybe we want to use something like lasso or ridge. And so linear mixed models often do things like that. They'll use all genotypes or all genotypes on a different chromosome in the SNP to predict the phenotype. They residualize it out, hoping that removes any sort of phenotypic stratification within the data, and then they regress the SNP, or they regress the residualized phenotype onto the SNP. And I can talk more about this later if you'd like, but that's the basic idea behind these linear mixed models, and that's why they're working. They do the same kind of prediction and residualizing, hoping to residualize out any of these confounds. So, concluding, stratification is one of the most important sorts of confounds that we'll worry about. The best way to eliminate it is to control for parental genotypes, and you can impute them using Alex's method that we're going to learn about next week, I think. And so that's going to be the most effective way to remove it. There's stratification. It can be there even if your sample is racially or ancestrally homogeneous. So you still need, you don't get a free pass. And there's a bunch of tools that exist to assess if there is stratification in your results, and also to remove it. But even those tools aren't perfect. So be cautious. Think about the applications. Think about how sensitive your applications are going to be to these kinds of concerns. Oh, this is just a supplementary slide if you want to learn about Frischbaug. OK, I'm done.
Even after adding like 100 PCs, we still see some regional patterns in like education PGA. So this PGA, so is this because those like sample size is quite limited? So those like 100 PCs, higher order of PCs, like perform very poorly. Then it doesn't really capture those regional stratification patterns.
Yeah, I mean, I don't think that we have like a concrete answer. I don't think we really know exactly why controlling for 100 PCs wasn't sufficient to account for the stratification that you see here. I mean, this is why I have a slide of like, here's some possibilities. And so I think it probably has to do with just the stratification being more recent, and recent stratification is difficult. Like it may show up in the PCs, but it'll show up late in the PCs, and the later PCs require very large. It has a combination, require very large sample sizes to get those well estimated. I think you had a question too, or did you not? Oh, I thought you were raising your hand before, but if you didn't, that's fine. Yeah.
I really should have asked this in the beginning, but we kind of went very quickly from like an interpretation of PCs into ancestry groups, and there's evidence that it maps out geographically, but just a priori, that's not really what I would expect necessarily, right? So I'm just wondering, ultimately the PCs are just like the eigenvectors of the design matrix.
Yeah.
So especially for higher order PCs, it's not really clear to me that they should be given this like geographic or ancestral group interpretation.
Yeah. So there's kind of two things. So one is just sort of an empirical observation. So in the Europe I showed you that it's really clean. If you have like diverse global data that you kind of know where the people are from that you analyze, you actually see pretty clean clusters based on kind of what continent a person's from. And so empirically, it seems to do a pretty good job of capturing kind of big continental kinds of patterns. And so that's one reason that we think like, oh yeah, this is working. Seems to be working okay. It also is like if you were to model, it's like a population geneticist. You might say like, imagine you have one big population and you build a wall between them, and then so individuals on either side of the wall are mating randomly among each other, but there's no crossover across the wall. And then you watched how the allele frequencies evolve between these two groups. Like you can do some random matrix theory and calculate how well the PCs are going to pick out these two populations. And over time, PC1 is going to almost perfectly discriminate between these two groups. Like I don't know how many generations are required. You have to PopGen person. But you can write out these simple models, and the PCs do exactly what we want them to do. The world is more complicated than that, right? Like the wall has holes in it, and people like escape from side to side sometimes, and things like that. And so, and in those cases, I think even in those cases, if you make some simplifying assumptions, PCs still do a pretty good job of identifying, what fraction of your ancestry comes from which side of the wall. But then once you start getting really complicated, people are moving all over the world at different times. It's not clear. PCs don't seem to do as well, which is why we went back to this. Why we're saying that PCs aren't sufficient to capture everything. But for kind of simple types of structure, and it seems to do pretty well. Dan? Oh.
I was just going to say, you do actually have to do various things before you estimate PCs to make sure you get PCs that are representative of like ancestral groups, right? Like,
are you talking about choosing SNPs and things?
You
Or?
have to remove SNPs that are in local LD because otherwise it'll just pick up like blocks of SNPs that are correlated with each other. You also have to remove relative pairs, so it's not exactly the same as the eigenvectors of the genotype matrix as you would do GWAS on, because you're actually filtering out various other things that would be picked up by the eigenvectors of the full genotype matrix.
Yep. Dan?
I was going to say, I think in theory you expect the PCs in these simple models to do well at picking up ancestry differences that have been around for a long time.
Yeah.
Right? So we don't expect them to do well necessarily with recent ancestral differences.
And you can think of families, right, as population stratification, like as super recent stratification, right? If you have parents and they have a bunch of kids, PCs aren't going to capture like, I mean, I guess if you have really, really big families, but in general, they're not going to capture things like that. But they maybe would capture like fifth cousins. Yeah.
Would you say that as people migrate around the world much more, and perhaps their ancestral backgrounds doesn't correlate as well with the exact environment they were nurtured in, PCs would not be as great to control for population stratification if we run the same GWASs in, say, 50 years from now?
Yeah, so the question is about like if the world becomes kind of more mobile, and so people are just kind of moving all over, and mating is slightly more random than it is now. It's sort of a way of saying what you just said, I think. Then will PCs still be useful or not? And I mean, it will take a little bit of time, right? I mean, you can think of stratification as kind of random mating, or assortative mating, right? I mean, I don't think 50 years would be enough to erase the stratification that exists already. But like, yeah, I mean, sure. If eventually everyone is totally mobile and who you have children with is no longer a function of like where you're born, or I guess what you'd need is for it not to be a function of anything, right? If it was just totally random, like just pick people off the street, then PCs wouldn't help. But you also wouldn't there would be no stratification in that case. But as long as there is any kind of stratification in society where it's less likely that I'm going to have kids with this group of people than this group of people, then PCs should capture that as long as it goes on long enough. It needs to be kind of long-run stratification like that. I'm not a population geneticist. I should also caveat that for the people at home. So I might be wrong, but that's just my sense. Should we have lunch? Okay. Thanks, everyone.